The overall goal of this experiment is to understand the direct metabolic regulation of ER-alpha in breast cancer using genome-wide transcriptome, cistrome, and metabolome data.
This method can help answer key questions in the nuclear receptor and cancer biology fields, such as the role of certain transcription factors in regulating metabolism to contribute to cell aggressiveness. The main advantage of this technique is that it gives a global picture of gene and resulting metabolic regulation events. Demonstrating the procedure will be Yiru Chen, a postdoctoral fellow from my laboratory.
After culturing MCF7 cells according to the text protocol, harvest the cells by adding 1 milliliter of RNA isolation reagent to each well and incubate at room temperature for 5 minutes. Use a pipette to collect the cell lysate and transfer it into 1.5 milliliter tubes. To isolate the RNA, add 200 microliters of chloroform to 1 milliliter of cell lysate and vortex for 10 seconds. After incubating at room temperature for 5 minutes, centrifuge the samples at 16,000 times G and 4 degrees Celsius for 15 minutes. Following centrifugation, carefully transfer the aqueous top phase to a new tube. Add 500 microliters of 100% isopropanol to the aqueous phase and mix by inverting. Then, incubate at -20 degrees Celsius overnight. The following morning, centrifuge the samples at 16,000 times G and 4 degrees Celsius for 10 minutes.